in the last class we started looking at resonance assignment of proteins. As I mentioned, these are the two steps which are involved in any protein resonance assignment. Number one, you have to identify the spin systems. So, this is basically called as assignment of spin system or identification of spin systems. So, how do we identify a spin system? We identify based on the pattern of chemical shifts in that spin system, like we saw in Toxi. Similarly, if you have a carbon chemical shifts, you can use that to identify that the spin system is there or not. So, basically here it means that suppose I have 4 lysines, 6 arginines, 7 aspartic acid and so on in my protein, I need to figure out whether really those number of amino acids are there in my spectrum or not. So, that is the concept of spin system identification, where we try to find out what are the amino acids which are supposed to be present, are they really present in the spectrum or not. Once we identify the spin system, the next step is to put the spin systems in the right order. So, this is important here. Remember, we have only identified at this stage that there are 4 lysines, there are 4 arginines, there are few aspartic acid and so on, but we have not arranged them in the right order. And how do you arrange them in the right order is the whole idea of sequence specific resonance assignment, which we will see now in this class. But the whole idea is that we need to know the sequence of the protein, so that we know how to order them, because if we do not know the amino acid sequence, we will not be able to put them in the right order. So, therefore, first we have to identify and then put them in this correct order. So, now this assignment of proteins actually typically happens in 4 different stages or 4 different steps. The first step is that we assign for every amino acid the proton nitrogen, that is amide proton or basically the backbone amide proton N15, C13 alpha and C13 beta, assign sequence specifically for every amino acid in the protein. The next step is, after we have assigned this, we assign the carbonyl chemical shifts, because this once you know which amino acid has which carbon, which proton amide alpha, we can then go to carbonyl chemical shifts and that are assigned again for every amino acid. So, once this is accomplished, then we go to the side chains. For side chain, we need to first identify the backbone H alpha and the side chain H beta chemical shift of each amino acid. So, this step is important because this is our entrance into the side chain. So, up to here the first two only concerns the backbone chemical shifts, but now we are entering the side chains. So, for entering the side chains, you need to have first the alpha and the beta protons of every amino acid. Once we have that, then we can assign the rest of the side chain, that is the gamma, delta, epsilon protons and similarly gamma, delta, epsilon carbons. So, that is the last step, and after that we can consider that the assignment is complete. So, these are the 4 steps. So, let us see how we go one by one in proteins through each of them. So, we will first start with the backbone chemical shifts. Now, in a given 3D NMR spectrum, we have to, as we have been discussing, we have to assign them in form, we have to analyze them in the form of strip plots. So, this is example of a strip given for a 3D experiment. So, typically what we do is we start from a base, that is projection. So, the base projection typically is N15 HSQC where each amino acid here is one peak. So, what we do next is we take this peak and then go to the 3D, and then in the 3D we look at the chemical shifts in the third axis. So, this is how we will see how we will analyze and assign the protein. So, now we looked at the very simple experiments triple resonance earlier, that is HNCA and HNCOCA. So, we will use this experiment as a illustration of how assignment is carried out to start with, then we will move on to more difficult or complicated experiments. So, first is that HNCA, what is the information do we get? For every amide nitrogen and proton chemical shift, that is for every residue, every amino acid which has this chemical shifts, in the third axis, that is the third dimension, we get the C alpha of the residue I minus 1 and the residue I, okay, for the same amide proton chemical shift. So, that is shown here in the strip format. Now, how do we know which is I minus 1 when which is I? That information comes from HNCOCA. So, this is shown here schematically. So, we have NH, and what we have done is we have correlated the HSQC. So, this is each strip here, this strip here and this strip here is basically an HSQC pair, because nitrogen and amide correspond to one pair of this residue amino acid. Now, for each amino acid in this experiment we are getting I and I minus 1 for each pair of nitrogen proton chemical shifts, we are getting I and I minus 1, that is we are correlating to this and this. Okay. So, basically that is what we saw in this here, we are correlating in HNCA the chemical shift of the nitrogen and proton with the C alpha of I, which is I minus 1 residue, and C alpha of ith residue. So, this kind of a connection, that is where you connect to both plus its own chemical shift and the previous one, is very important in assignments. This is something we saw in the case of Toxi as well in the NOSI for a peptide which I showed in the last class, that if you take a peptide like alanine, glycine, there also we saw that in the NOSI what helped us to assign was the inter amino acid connectivity. Similarly, here this is the inter amino acid, between the two amino acid connectivity, which is very important for assignment. Without this connectivity we cannot assign, because only having the self has no big value for assignments. We will see that shortly now. So, now say for example, for every peak in HSQC I will basically get the correlations to I and I minus 1. So, I can move this to the previous amino acid to I minus 2, I minus 3 and so on. I can also move this side I plus 1, I plus 2 and so on. How do I do that? I will do by joining or connecting this C alpha now with the next NH here, because remember this C alpha can be connected to the next NH, because it will be the I minus 1 for that amino acid. Similarly, this C alpha can be connected to this NH, because it is the self. So, remember the nomenclature, we will call this as self and this as sequential. Okay. So, let me write down this for to be sure. So, this we will call it as self, that is I, and sequential, which is I minus 1. So, this is the nomenclature used. Now, I can go to, for example, this peak, let us say any arbitrary peak in the spectrum can be this particular amide. So, that gives me correlation to this. Now, I do not know which amino acid is this. I have just started with some arbitrary amide proton chemical shift in the HSQC. Now, if I go here, I can now see which this peak, which peak here, which of this peak here corresponds to this. How will I know that? That peak, whichever is that peak, will give me correlations to this C alpha. Okay. So, this C alpha is basically now shared between this and this, because for him, for this particular case it is I, and for this particular case it is I minus 1. So, they share this C alpha between I and I minus 1. So, I can therefore have a common connectivity here. So, basically this NH, once I find that it could be some peak here, let us say it is this peak here, which need not be nearby, it can be anywhere. Once I have found this peak, now I have found the I minus 1 chemical shift, but again I do not know which I number we are looking at, it is just any I. So, I am not saying that we already know what I is and what number is I, we are just saying some arbitrary I and I minus 1. Now, once I get I minus 1, it will give me correlation to I minus 2, because that will be the HNCA correlation. Now, again if I go further, I can get I minus 2 NH by correlating it to this C alpha, which is again from HNCA. So, I basically need some peak which corresponds to this NH in the HSQC, which will now give me correlation to this. We will see this again as we go further. Similarly, I can go on this side. On this side, I can again find a NH pair, this is one pair which will be some peak here, which will give me correlation to I and also I minus 1. So, you see this is a common connectivity here now, it is shared between I and I plus 1. So, I can find now some NH somewhere here, which could be anywhere, let us say it is this NH, and that NH turns out to be such that it has a connectivity to this C alpha, which was connected to this NH. So, these two now are connected to a common C alpha, and that could be I plus 1 therefore, and this is now I by plus 1. Now, this further will connect to I plus 2 and then I plus 2 will go to I plus 3 and so on. So, you see what we have done here, we have connected inter amino acids, 2 amino acids by this common NH. So, in other words, one C alpha is shared by 2 NHs. You can look at it in that way also. Okay. So, this is basically how we will see. Now, we will go further and see how assignments are done by connecting one NH to another NH through C alpha. So, this is the basic point here. We are connecting two neighboring amide proton pairs through their common C alpha to which they are correlated. Okay. So, we will get the complete stretch, we will be able to connect a stretch of amino acid from I minus 2 to I minus 4 or so on to I plus 1 to I plus 2 and so on. Remember, this connectivity does not go from minus one end to the other end of the protein, it will only go to 5 or 6 amino acids, because after 4 or 5 roughly you may encounter some breaks. And what could be those breaks? The breaks could come because there is a missing peak in the HSQC. Remember, I said in HSQC you never get 100% peaks for a protein. So, if you expect about 100 peaks for a protein, you may probably get only about 80-90%. So, those missing peaks are basically those peaks which are simply do not appear in the spectrum. So, if they do not appear, there is no way for us to identify that amino acid. So, for example, it may happen that this NH is completely missing in the spectrum. It does not mean this is missing, it will mean only this is missing. This could be missing because of various reasons like exchangeable fast exchange and so on. So, it could be possible that this NH is missing, and therefore, we are not able to continue further this side because there is no information on this. Similarly, here we may go up to I plus 2 and I plus 3, and then again we may find a missing peak, and then again we will not be able to proceed further. So, we would have assigned from I minus 2 to probably up to I plus 3, which is about 5-6 amino acids. Okay. So, this is how we go in batches of 5 and 6 amino acids along the protein sequence and try to complete the assignments. Now, one more thing is another reason break could happen is because of proline. Proline does not have NH proton. Proline is a special amino acid which does not contain NH. So, if that is the case, therefore, when I go to the next amino acid, I will not get any peak in the HSQC because there is no, the proline cannot have any peak in the HSQC. There is no NH pair in the proline, and therefore, that constitutes a break. So, now let us continue and see how this assignment is done. So, you can see this is the basal plane projection of a 3D HNCA. So, in this what you are seeing here is a HSQC spectrum of some protein. This is a very schematic drawing, and you can see each of this peak corresponds to one amino acid. Now, on this plane you get the 3D of HNCA, so which will look something like this. Okay. So, let me write down, we are looking specifically at 3D HNCA. Okay. Now, in 3D HNCA, for every NH peak we get I and I minus 1. Again, remember, we do not know which is I minus 1. So, we need HNCOCA, but let us say we have HNCOCA which is not shown here, but based on that let us say we have identified that this is self in this particular case and this is sequential, like that for every case. So, we will not we will assume that we know which is I minus 1 because we have used HNCOCA which is not shown here. So, let us start from some NH, any arbitrary NH here. So, let us say I start from here, let us call it as I. When I start from here, I will get 2 peaks which is I and I minus 1, that is C alpha. Now, I will search in this whole 3D cube where this I now appears as I minus 1 to I plus 1. So, for I plus 1 something is I minus 1 is nothing but I. So, the sequential of I plus 1 is I. So, this I and I plus 1 they share this common chemical shift C alpha I. This is what we saw in the previous slide. In the previous slide, let me go backwards a little bit. we saw in the previous slide. So, if you look here, the I plus 1 NH and I NH, they share or they correlate to I C alpha. Similarly, I NH and I minus 1 NH correlate to I minus 1 C alpha. So, they have a common connection. So, same thing is shown here. From I go to I and I plus 1 NH peaks, they share a common C alpha I. Now, in this line, if I go top, the remain this is I plus this is I. So, this will become I plus 1, because I plus 1 and I is what you will see on this peak. Now, this peak will be shared with I plus 2, like we saw in the previous slide. Similarly, this I plus 2 will be shared with I plus 3. So, like this we can basically go along the chain by simply connecting the common C alpha chemical shifts. Okay. So, that is how basically assignment is done, but as I said, after this you may probably get a break, you may not be able to see I plus 4, because I plus 4 may be missing in the spectrum, because of either peaks are weak or there is a proline. I plus 4 could be proline. So, we do not know what is I here. We started from some arbitrary I and then walked along the backbone. Similarly, I can go to I minus 1, I can start from this will be shared with some other I minus 1 and so on. So, you can see this is how the connectivity is shown in this picture. So, you can see that for a given this is a strip plot format, in a given for a given NH and amide proton pair, I will get I and I minus 1. Here we have shown filled circle here and open circle for I minus 1. Remember, this distinction comes from HNCOCA, which is not shown here, but I am assuming that we have recorded an HNCOCA, we have already looked at it, and then based on that we know which is I and which is I minus 1. Now, this I is shared with I plus 1, that is what we saw in the previous slide, and this I plus 1, this I is shared. So, this when big this is I, this the other peak in this line becomes I plus 1, because we are on the amide of I plus 1. This I plus 1 now is shared with I plus 2, and I plus 3 2 is shared with I plus 3 and so on. So, this is how we walk along the backbone. This is called sequential backbone walk. It is a very common terminology which is used in protein assignment, which is we will be talking about more. So, now if you see what we have achieved in this, basically this whole procedure is that we have achieved actually the second step. We have identified the sequence in order, okay, but we have not got the spin systems. Why? Because look at it here, I am only talking about some I do not know what is this I, what spin system is this I. Is it a glycine? Could this be a lysine? Could this be an arginine? Could this be an aspartic acid? We do not know any of that information from this exercise. So, this whole exercise is only giving us a connectivity pattern between amino acids. So, this is one problem with using HNCA only, and HNCA and HNCOCA of course, is that we do not get the amino acid type information. We do not come to know which amino acid is which peak, because we just start from a I, we assume some peak C alpha, but C alpha has no information of what amino acid it is. Many C alphas of many amino acids, they all look alike, means they come in the same spectral region. So, therefore, I need to achieve this assignment. So, this is what is written here, that with C alpha chemical shifts alone you cannot achieve the identification of spin systems. So, therefore, HNCA is not so useful for assignment, in the sense it does not help us to get this part. So, how do we then improve this? This can be improved with using HNCA-CB. So, this is again the same thing is shown here, that for every amide proton in HNCAs we are sharing the C alpha. For example, HI and NI, which is one peak in HSQC and this is another peak, they share the common CI, C alpha I. Similarly, I plus 1 is shared between these two, here I minus 2 is shared between these two and so on. But the problem is, how do we find out where does this stretch belong in the sequence? So, we have connected a stretch from I minus 3 to I plus 2, but we have no clue, no information on which amino acid type is this. Like for example, is this arginine, is this glycine, is this isoleucine, some kind of amino acid. Information is not there in this assignment, in this connectivity. Therefore, we need to know what amino acid it is before we can say that we have completed our assignment. For that, we need to go to another experiment, that is called HNCACB and HNCOCACB. So, this is what happens in HNCACB and HNCOCACB. We have seen this here in HNCACB, the amide pair, the nitrogen proton pair correlates, means connects to C alpha and C beta of I minus 1 and C alpha and C beta of I. So, you see the extra thing is happening is C beta, the C beta here is and C beta here, and this information was not present in HNCA. So, when we were doing HNCA and HNCOCA, we only went up to C alpha, but not to C beta. So, here we are going further one step to C beta. So, in HNCOCACB, we get information of C alpha, C beta of I minus 1. Remember, this residue is I minus 1. So, be like similar to HNCA and HNCOCA, we can now say like this, that for every given amide proton pair, that is every residue I, the self chemical shifts are C alpha, C beta, and I get sequential chemical shift these two. Okay. So, this now I have one extra chemical shift which I can use for connectivity. So, this is why the reason these experiments are important is because C beta carries a lot of information of amino acid type. We will see that shortly, and therefore, we can identify the spin system which was missing in the HNCA. So, how does these two work? It is similar to HNCA matching. So, you can see here in this schematic that in a strip plot format, suppose I start from some arbitrary I, okay. So, arbitrary means any I, there I will expect to get 4 peaks in HNCACB. This is something you might be now familiar with from the previous part where we looked at 3D NMR. There we saw that you get 4 peaks corresponding to C alpha and C beta. So, now you can see that these two, which are now self and sequential, are colored separately. So, shown here yellow is self and sequential is red. So, these two now sequential means which are I minus 1 can be identified based on this spectrum, and whereas the self is different. Now, this self will be shared common with I plus 1. This is something we saw in the previous slides, that I and I plus 1 amino acids share the C alpha I and C beta I. Now, these two become the sequential for this amino acid. Okay. So, for this amino acid, now that is sequential, it is I is sequential and I plus 1 is shown here. Now, I plus 1, if you go further, you will find another match to I plus 2. For I plus 2, these two become sequential. So, you see the self is becoming sequential when you go this side, and the sequential will become self when you go this side. So, self and sequential are interchangeably changing because whatever is self for this amino acid will be sequential for the next amino acid. So, this way we can connect now 2 chemical shifts and continue the assignment. So, what is the assignment we are doing? You are connecting to find a connectivity, a stretch of amino acid which are connected. So, you see in the advantage of this part is that we not only get this, we also get this information. We are getting both that information, that is assignment of identification of spin system also comes from this C beta, and the connectivity also comes from the C beta. So, how does the spin system come? Let us see in this particular slide. So, you can see here that what we have done, we have I minus 2. So, we have connected, we started from an arbitrary H and an I in a protein, some peak in HSQC, and we went this way and this side. So, when you go I plus this side, you will see we share this 2 chemical shifts between the neighboring amino acids. So, very important point is neighbors. These neighbors share have a common correlation to these chemical shifts. Similarly, if I go to I minus 1 side, I will be sharing this for these two, C I minus 1, C beta I minus 1, and similarly between I minus 1 and I minus 2, they will be sharing this chemical shift in common, that is C A I minus 2 and C B I minus 2. So, like this I am forming a connectivity pattern or connection between neighboring amino acid. But again remember, we have only connected, we have not found out which amino acid it is, we have simply formed connection. So, how do I find out now which amino acid is this, which is this, which is this, and so on? It is based on this C beta value. Okay. So, written here C beta chemical shifts has a very good information of amino acid type. So, let me show you a statistical analysis which was done long ago, and this is the result of that analysis. Here you can see this is a 2-dimensional plot with C alpha on the x axis and C beta on the y axis. So, you can see that amino acids cluster into different groups based on their C alpha and C beta. So, for example, let us start from here. Alanine is very distinct chemical shift as far as C beta is concerned. Look at this axis which is on the vertical axis, C beta comes in this zone. This was based on thousands of chemical shift from database. So, you can see this is a range of chemical shift for alanine. Similarly, these groups of amino acid lysine, arginine, glutamine, so on, they come in this category, they come in this zone. Okay. Similarly, this zone blue color belongs to these amino acids, and a few of these valine and isoleucine, they are actually very unusual. Their C alpha comes, this is now looking at C alpha, comes somewhere around 65 ppm. Okay. So, this is unusual special case for valine and isoleucine. Now, if you further go down in C beta chemical shift, see, you see there is a gap here, there is no C beta here, mostly it is C alpha, but C beta there starts around 60 ppm for serine and then further down for threonine. So, serine and threonine also can be separated. So, you can see here how nicely the C beta chemical shift gives us information of what amino acid type it could. Of course, here yellow means all of them could be here. So, it does not tell us between these amino acids. Similarly, blue color is full set of these amino acids, we cannot distinguish within this set, but still we are able to get some information which was not available from C alpha. So, a combination of C alpha and C beta is helping us to achieve that information. So, once I have that information, I can probably predict, suppose the C beta of this case was 16 ppm, I can say that could be alanine, because that is what I saw from the previous chart. If it is 32 ppm, probably it is histidine based on database. Similarly, here it may happen that this C beta is 0, means there is no chemical shift of C beta for this amino acid or this amino acid, which means that it is a glycine, because glycine does not have C beta, glycine is only up to C alpha. So, we could get a glycine. Similarly, here it could be that it is 67 ppm, and based on the theory this could be threonine, like that. So, based on this database and analysis, statistical analysis, we can predict or guess which amino acid type it is based on the C beta. Okay. So, once you have guessed or predicted that, you can now see that I have connected a stretch of 6 amino acids. Okay, we have connected a stretch which lie one after the other. That means, if this is I minus 2, this is I minus 3, this is I minus 2, I minus 1, so on. So, they are continuous. Not only they are continuous, this is a specific set of amino acid. Now, can we try to find that in the sequence? So, I am trying to find these amino acid in the sequence in this order only. The order is coming from here. So, I have connected in the right order. After connecting them in the right order, I have found the chemical shift type, amino acid type of these spin systems, and now I am trying to find where they can belong in the primary sequence. So, remember again, we have given this sequence, we already know this sequence. So, we are trying to map. So, this is called mapping. It is a very important word in assignments, mapping. So, we are trying to map the stretch of amino acid that we have assigned on the sequence, and then if there is correct assignment, if this what I have done is correct, then I should be able to find that sequence in my protein sequence also. So, my protein primary sequence should contain this stretch of amino acids which I have connected and assigned. If it is found, then I can say that yes, my assignment is over for these amino acids. Again, remember, we are not talking about the remaining ones, only this 4 or 5 or 6 amino acids we are assigned now. So, we will now see how we can continue this assignment in the next class, and we will look at how we can complete the full protein chain starting from one end to the other end.